So now it is my pleasure to introduce again to the stage, and I'm curious to see if he put his tie back on. Udit Batra, the CEO of MilliporeSigma. Thank you so much.
Thank you. Thank you. It's a tough act to follow, and I can see from the crowd that politics is a lot more interesting than breakthroughs in science. But that said, for those of you who are here, we have a real treat. We have a panel that's going to talk to us about cell therapy, one of the breakthroughs in science today, and something that has the potential to change how medicine is practiced. Please take a seat. We have a few chairs, and it's rather cozy, but I think we will manage. Before we go into the panel, we wanted to show you a video so that we are all on the same page on what cell therapy really is. Can we have the video, please?
The immune system is the body's defense against infection and cancer. T cells play an especially important role in the human body, designed by nature to recognize and kill diseased cells, whether infected by a virus or a cancer cell growing out of control. Unfortunately, the human immune system isn't perfect. While the scientific and medical community have made great strides in finding new therapies, there are rare forms of cancer that don't respond to any kind of treatment. Enter chimeric antigen receptor T cell therapy, CAR-T for short. CAR-T is an exciting new personalized cancer treatment that's showing tremendous promise. Those T cells I just mentioned, they can be genetically engineered so that the body's own immune system can kick into high gear to fight cancer. Creating a personalized cell therapy can save a life. But the process to manufacture CAR-T cells is time-consuming and has risks along the way. It takes about 30 days to re-engineer the cells and get them back to the gravely ill patient. It's a race against time. Let's take a closer look at a T cell's journey to become a CAR-T therapy. Step 1. Blood is drawn from the patient at a hospital or clinic. Their T cells are separated, collected, and usually cryopreserved for transportation to a manufacturing facility. This journey could take up to 48 hours. Getting the T cells to the manufacturing facility is the first challenge in the process. Anything from bad weather to customs delays can mean the cells never make it further in the process. Step 2. The T cells are sent to a cell therapy manufacturing facility where the sample is prepared and the cells are selected. A patient's immune system, compromised by the cancer or prior chemotherapy, may mean that not enough healthy T cells can be captured. If that happens, you can't make the CAR-T therapy. More T cells will need to be collected from the patient, which will delay treatment. Step 3. In the lab, genes that recognize specific cancer cells are inserted into the cells using an inactivated virus. The number of patients' genetically modified T cells is expanded by growing cells until there are millions of them, transforming them into CAR-T cells. This can take up to 2 weeks, depending on how fast the cells grow. The CAR-Ts are then frozen and sent to the hospital where the patient is being treated. Step 4. But wait, before we even go to Step 4, we should talk about that inactivated virus, also called a viral vector. You can't re-engineer cells without them. Viral vectors are harmless viruses, commonly used by molecular biologists, to deliver genetic material into cells. Viral vectors are what transform a T cell into a CAR-T cell, but to do that, you first have to manufacture them. Here's how it works. Small bits of DNA, called plasmids, instruct production cells how to make the virus. These plasmids and production cells are mixed and then used to manufacture the virus in large batches. Each batch of virus needs to be tested for purity and safety before it can be used in the CAR-T manufacturing process. The manufacture of viral vectors needs to be complete prior to the manufacture of CAR-T cells. This is a long and complicated process within the supply chain. In fact, the manufacture of viral vectors can take up to 1 year. Clinical supply needs to be ready before the patient needs it. Step 4. The new re-engineered cells are concentrated and placed into a formulation that is compatible for infusion. This step usually takes less than 24 hours. We are more than halfway through this 30-day process. The clock keeps ticking. Step 5. The CAR T cells need to be tested for sterility, quality, and safety prior to release. These tests can take from 3 days to 3 weeks, depending on the methods used. Once complete, cells are preserved in a manner that assures their safe transportation back to the hospital where the patient is being treated. This takes 24 to 48 hours. Step 6. The CAR T cells are now ready to be administered to the patient. Once infused into the bloodstream, they multiply in number. These attacker cells can recognize and kill the cancerous cells that have the targeted antigen on the surface. Testing can be a lengthy process, and there is the risk that the cells get contaminated during manufacture, rendering them unusable. 30 days, needle to needle. A race against time to save a life with personalized cell therapy.
Patiently listening to that video, but to simplify it, I was trying to explain this to my 10-year-old son, who summarized it rather simply. He said, "Dad, cancer is like a super spy, and these T cells can't find it. You need to just give them a bit of help and guided missiles to find these cancer cells, and then you can destroy them." This whole process that we described, I think, is simply summarized in the words of a 10-year-old. But we have highly sophisticated panelists who will tell us a lot more about how difficult it is to make this therapy available to many people. Before we get started, I know they're super excited about the therapy itself, so we'll ask them to just briefly tell us their names, not go through their CVs, you can read them, and just tell us why they're excited about this wonderful therapy before we discuss its challenges. Start with you, Dr. Levin.
I'm Bruce LeVine. I'm a professor in cancer gene therapy at the University of Pennsylvania. Our team developed CAR T therapy directed against B-cell leukemia that was recently approved August 30th by the FDA and developed by Novartis. You asked, why am I excited? I'm excited not only because it's something that I've been working on for 25 years, but I've seen pounds of leukemia killed by patients' own immune cells that were engineered to kill their cancer. I've seen patients who have been told they should plan to go to hospice now participate in patient advocacy, start foundation to support cancer research. It's a true personalized therapy, and the other panelists can share their stories. David.
Yeah, I'm Dave Williams. I'm the Chief Scientific Officer and Senior Vice President for Research at Boston Children's Hospital. And I started in the field in 1982 as a postdoc at MIT, working on trying to develop the viruses that ultimately have been used in human gene therapy. In those days, it was work in mice. Those days it was working in the mouse, but we've adapted that over the subsequent 30 years, obviously, into the kinds of success that you just heard about. We, in our program, work in two distinct areas. One is correcting children who have genetic defects of their disease, and the other is the application of CAR T cells in our children with leukemia. And the exciting thing is just having the patients in front of us that we can offer these unique therapies for who have often deadly diseases. And so the joy I get out of as a clinician taking care of them is offering these very unique therapies in areas that give them hope where before there wasn't any hope.
I'm Marcella Moss. I'm an oncologist and the director of cellular therapy at Massachusetts General Hospital in our cancer center. I actually trained with Bruce LeVine at University of Pennsylvania and the whole team there. And I'm really excited to be able to have a cell therapy service at Mass General because that's how far this therapy has come. But for me, the most exciting part is, of course, treating the patients. When we started this and when I was in college here at MIT, it was a pipe dream. It was science fiction to think that we could use viral vectors and genetically modify cells and put them back into patients. Not only that that would be safe, but that it would actually work. And so it's really been amazing to watch the field go from not being able to use the word cure ever in oncology to now thinking about whether that's a real possibility, and patients now have hope that they didn't used to have.
Hi, I'm Jeff Hodge. I'm the Chief Technical Officer of Unum Therapeutics. We're a Cambridge-based company just up the red line at Alewife, and we're working on a next-generation CAR T cell that hopefully has opportunities for better safety profile and the potential to get into some other indications. I'm excited. Part of the reason I'm at Unum Therapeutics is that my daughter had ALL, and she was treated with traditional chemotherapy, and she was cured more than 10 years cancer-free, which is wonderful. But chemotherapy is a really, really brutal treatment for cancer, and even the survivors come out of it very, very damaged. And of course, not everybody does survive. And I'm really excited because cell therapy has offered an entirely new mechanism that we can potentially exploit to try to defeat cancer.
Hello, everyone. My name is Shashi Murthy. I'm a professor of chemical engineering at Northeastern University, where my lab develops and commercializes technology for the manufacturing of these personalized cancer therapies. I'm very excited. I'm very excited because this is really just the tip of the iceberg, in my opinion, in terms of a paradigm-changing approach in medicine that will extend beyond cancer, I think, but then also result in innovations in manufacturing and technologies that will enable significant enhancements in the way we treat patients, but then also create new technologies and new ways that people are employed and engage themselves.
Hi everybody, I'm Dave Backer, and I work with MilliporeSigma, and my part is really on the right-hand side of the chart and has been on the viral vector manufacturing. And in the past years, viral vectors were used as a direct injection. The virus was trying to get the gene through true gene therapy. And sometimes it works, and many times it didn't. And it wasn't until you could get the cell, the specific cell, and genetically modify that cell that we really started to see some tremendous successes. And so it's to the group here and the scientists that have developed these technologies that are laborious right now, and we have to work on how to streamline that. So I think the vectors are a critical part of just trying to get that gene into the right place. And from a personal standpoint, I have a daughter with a monogenic neurologic disease. So I want to find a way to get to that neuron. It's something that we can't do right now, but it's the success that we're seeing here, and it's really incredible the changes that have happened just in the past 5 years, where we're really seeing all these genetic diseases where we could find an approach.
Dave, and as you can see, there's a lot of potential for cell therapy. And I'm sorry, there's an echo on the stage. We can't really tell if you're hearing us or not. If somebody in the back who doesn't hear us can just go, we will try to move on the stage to prevent the echo. I think it's basic physics, but I think we can figure it out if somebody helps us in the back. Let's start. I think there are several challenges with such a breakthrough therapy, and it's in its early stages. Let's start with you, David. We know that this is a bespoke therapy. This is a therapy that is individualized to individual patients. What are the challenges with such a therapy? Let's start with you, and maybe Bruce, you'll comment on it as well.
Well, I think I'll say that there's two extremes in my experience. On the one hand, as I said, we're now offering patients life-saving therapies, and so we have the reaction to patients who have multiply relapsed leukemia, who essentially have no therapy currently left in the armamentarium to treat them. They're extremely grateful for this opportunity. On the other hand, this is such a complicated manufacturing process, and the time wait is frightening to people as they're sitting there with their young child, boy or girl, waiting on the therapy. And it was mentioned in the video, there are manufacturing breakdowns. So we've had the experience in our trials of having patients ready for therapies, but not being able to give them because of the manufacturing process didn't pass QC. So on the one hand, it's a wonderful new set of therapies. On the other hand, with biopharma just now coming into it in a sense, there's a lot of room for improvement in the processing and hopefully improvements in both the quality and the time frame it takes to get the product you need for the patient.
A lot of room to standardize before we move further and make it available for many, many more patients. Bruce, did you want to add something?
Yeah, so beyond it being one lot per one patient and all the variability that goes along with that, everyone's cells are different. We do have the science. We know that it works. We know that we have patients in our center that are 7 years out free of leukemia. The first child is 5.5 years out free of leukemia. But we are in the Model T stage of developing this technology. We need to get to the Model S stage, or the Prius out there that's hydrogen powered. Now that took some time, but I think with more folks like Shashi coming into the field, we have tools and equipment being developed that can automate this process. It will always be an autologous cell therapy bespoke process, but there are companies working on off-the-shelf engineered cells. Now there are challenges there because I can't give you my liver without it being rejected. There are ways that we can cloak those cells, but for the time being, they're short-lived. They could be a bridge to another therapy for patients from whom we can't generate their own cells.
Both improvements in the process as well as the cell line that we use to inject patients and treat them. Two rather obvious and simplistic things as it seems on surface, but highly difficult to implement. As you look at that chart, it's at least 3 different circles, and there are 3 different institutions that are working in collaboration handling the patient cells. You start at an academic medical center, you go to a centralized manufacturing facility, and of course, Dave Backer and his team and many like him are supplying what are called the viral vectors. In this highly disaggregated system, what do you see? This is currently the state of the art. What do you see as the pros and cons? Let's now start with you, Bruce. We'll move on to you, Marcella, after that.
Well, I'm going to take the liberty of changing your question a little bit, which is, what are we going to improve? We're going to improve methods of gene delivery. We're going to shorten the time of the culture. We're going to have improved ways to direct the cells, turn them on and turn them off in conditional fashion. You might call it the Google self-driving car of T cells. We can tell them what organ to go to and what organ not to go to. So all of these are in preclinical development. Some iterations of them have made it to the clinic, but the speed at which we're able to accelerate the development is increasing because of the clinical results. There has been investment in the sector, new companies springing up in the space, engineered T cells, engineered NK cells, and then companies like MilliporeSigma and others investing again in the tools and equipment that can engineer these therapies.
Marcella, did you want to add to this?
Sure. So I agree with everything Bruce has said. Definitely shortening manufacturing times and getting a more consistent product from patient to patient, and knowing that that product is functional rather than having to wait after it goes into the patient to see if it was really an effective product, I think is going to be very useful. I think that things that we can do to try to shorten the time of manufacturing, but also just the cost of goods. There's a lot of reagents that are sort of specialized, a lot of tools and ingredients that are only available for certain laboratories to be able to make the cells would be very useful, because we have a lot of ideas of how we can make the cells better, but there's limited availability of ingredients that we can put together to make that happen, and to make it happen quickly and to make it happen in patients rather than just testing it in test tubes and in mice.
But isn't there a conundrum? We have current state of the art that is somehow already been used to provide therapy to patients. And of course, we are developing new technologies across each of those steps and of course combining them. But as practitioners as well, isn't that posing a challenge, and can we not do something to reduce the variability that the patient has to experience?
So, it's challenging in diseases where we already know that the current methods work so well. It's challenging to change it, but there are certainly a number of diseases where there's definitely room for improvement. It would be wonderful to have standalone machines that you can take patients' blood and put it in, and then out comes a CAR T cell, within a week. Or something like that, and that was characterized, meaning that we knew all of its properties, what the active ingredient was, how well it was going to work, what its full potential was. And I think that that's in the cards. It's being developed by several different kinds of people, although I'll make a plug here that if you're an engineer and you know about bioprocessing and how to build these tools, there's a desperate need for it in the field.
We have one on the stage. Why don't we turn to him and start? Shashi, tell us what seems the hopes of medicine and really smart medical professionals is pinned on engineers. I'm a chemical engineer, by the way, as well. Again, the hopes are pinned on chemical engineers, so tell us what you can do.
So, there was an era before the internet, and now we're here where no two people's Facebook page is the same. So, it took a series of evolutions for that to happen. And that is what needs to happen in the area of bioprocess engineering with respect to cell therapy. I think we're really just getting started. I think there are significant opportunities for innovation ranging from the reduction in the number of steps associated with the development of therapies to making each step more efficient to then making instrumentation and chemicals and reagents customizable to either the type of therapy that's being manufactured or to the nature of the patient who needs a bespoke therapy. All of these are possible, but what's surprising is that there's very few people in the academic world working in this area of bioprocess engineering, and as a result, very few out-of-the-box innovations. But that is starting to change, and I think there's room for both optimism as well as new changes coming.
Give us an example, Shashi. That was helpful. Give us an example.
So one of the examples is the work done in my own lab, for example, with a company called Quad Technologies, which I created with a few of my students. So this company created a means to activate T cells, which is part of the process here, and to do so at a level of efficiency that is greater than what's possible with current technologies. And this came out of an effort, a long-standing effort to purify stem cells and related cells in very mild conditions. And so what we did was develop the reagents, but also the instrumentation around it. And so, I mean, that's really just one piece of a much, much bigger puzzle. But I think that, the combination of both nuts and bolts, pots and pans type of work, but also smart electronics, I think can make, a lot of improvements.
Can I jump in?
Yes, please.
I think it's interesting because what we're talking about with respect to the applications of viral vectors in human disease all essentially evolved out of academic laboratories. And so there's a real space for that. On the other hand, academic laboratories, because of the way they're funded, mainly by the National Institutes of Health, are generally not funded to do process development. And that's why there's not many people in that field. That's a perfect place for the combination of biopharma to work with academics to develop new processes and improve on, because in the academic centers, we have the experience of dealing with patients and how you deliver these things. And on the other end, you have really smart people at the basic science level, And in biopharma, you have great people like yourself who are engineers or from engineering background who understand how to apply processes to improve the reproducibility and the lack of variability that you need in a pharmaceutical, as opposed to the types of trials that we generally do.
That's really well put in terms of the collaboration that is required from actually many of the folks that are on the stage, and we're very fortunate to have all of you on the stage. Jeff, as a practitioner, what's your view on the topic?
Yeah, I think I share a lot of the optimism that you've heard from the other panelists. I do maybe want to temper it a little bit. I think there are probably, as Bruce said, we're kind of the Model T stage of this technology, and I think that we have a huge world of possibilities ahead of us. We're certainly going to learn more about making a better product, what makes a product better. We'll bring new technology to bear on it. We can certainly look at some easy wins like automation, not just for the process, but for some of the analytical assays that are very time limiting and time consuming. But I think it's maybe illustrative to look back at the advent of biotechnology, recombinant proteins, when we went from chemically derived small molecules like aspirin to some of the first proteins. The first antibody was approved by the FDA in 1986. And back in those days, people were happy to get tens of milligrams per liter of cell culture. And a decade later, people were getting hundreds of grams. And a decade later, people were getting grams. And now as we're pushing the fourth decade, people are pushing 10 grams per liter. Log order of improvement in productivity. This didn't come because of major innovation at each stage. It came as a cumulative innovation across the industry, a whole series of small changes and adjustments and improvements shared across the industry. So I think we'll see the same thing happen with CAR T therapy, cell therapy in general. We'll as an industry, learn an awful lot more about the process over the course of time. The FDA in 2003, not quite 2 decades after the first antibody was approved, declared that antibodies were now well characterized enough that they moved the regulation from CBER to CDER. So it was almost 20 years before this antibody that had been treating people was considered well understood enough to be a well characterized drug. Now a cell is infinitely more complex than a protein. So I do think we'll get there, but I also think we've got a long road. Fortunately, we're seeing amazing results with these drugs as we work on them.
I think that's the conundrum, right? The demand is so much higher than the state of the art can tolerate and can deliver today. And one of the bottlenecks, David, a question for you, is viral vector manufacturing. I mean, it's it's simplistic to say it's just an excipient or an ingredient in the whole process, but it appears that area is a bottleneck for many of the companies that we are serving.
The viral vector is a primary part of this equation because it is what is used to get the gene into these targeted cells. And I've been manufacturing these products, these Model T products, for 20 years. And in a traditional gene therapy standpoint or for immunology, we may make a batch that could give you up to 10,000 doses in a batch. But for these CAR Ts, we're talking about making the same volume, but because of the inefficiencies of the process and how many of those vectors are getting into the vector copies are getting into the cells, we may only have enough for 5 or 10 patients, as an example. So the technology is there, but we have to figure out how to make more of the virus to be able to scale it up, but also potentially how to condition the cells, so that you can get more, so the virus can be more effective. All of these things can help to reduce the bottleneck that we have. But I can tell you that the demand that has occurred just over a short amount of time to build facilities takes time, and then you fill them up. And we went from a 50% filled facility to a 150% filled facility in a matter of months almost. It truly was phenomenal how the demand shifted in this. And this is where technology really can change. And we've been playing catch up, and the other folks that have capacity, we're adding capacity, we're doing everything that we can to meet the demand. But it's tough.
Totally clear collaboration is a must. Bruce, did you want to start? And I know David also wants to make a comment.
Yeah, I'll say a little bit about another challenge that we're meeting currently. How many automobile mechanics do you think there were in 1905? Not many. So we're in the process of making more by building training programs, training modules, accelerating our training, but we need people to go into this field of bioprocessing, cell culture, cancer immunotherapy, so that we can employ them at the universities, at the institutes, at the companies, and not only develop these therapies, but manufacture these therapies so that we can treat more patients.
Must collaboration
I
and
would
training,
second that.
absolutely. David and then Shashi.
Yeah, just three comments to put into perspective the bottleneck. To follow up on what you said, so the virus vector system that we generally use for human treatments are called lentiviruses. We've just opened at Boston Children's a trial to cure sickle cell disease with lentiviruses. And the full production run that took a year to get will probably treat 2 patients. So this week I've been calling manufacturers around the world to, not for that trial, for a future trial, to ask when I could get the next slot, GMP slot.
Dave is the boss.
I'll give you my card.
And the answer was, we can probably slot you in March of 2019. So that's a real problem. And not so much maybe for big manufacturers like that can produce their own GMP facilities, but a lot of the drive of this, as I said before, is from small biotech and from academics. It's a real problem.
I think what we're hearing is even with the state of the art, each of the different steps have to be optimized, even if we are able to supply the demand that is out there today for patients that are in need. And I think we all need to collaborate on it. Shashi, one comment, and then we'll take questions from the audience.
I would just add that, you know, everyone ought to keep in mind that this is very hard work. You know, not only the science associated with developing the therapies, but also manufacturing them. And, you know, it's just something that, you know, you can't just make these cells in large numbers and just deliver them like, you know, you might M&Ms or iPads. So it's inherently hard and difficult, but I think a worthy challenge as well for everybody involved.
So I just want to say that, sorry, Bruce, but Boston is like one of the perfect places to do this because we have engineers.
Thinking the same thing.
He's from Philly.
Well, we'll get you over here, Bruce, at some point.
It's why we're called the Hub.
It's the HubWeek, and we have an invader. Any questions from the audience?
Hang on.
I think you need a mic. We can hardly hear ourselves.
Can someone try to characterize the difficulty of a production run that takes a year that yields 2 patients? Is it the yield of the modified cells that are the problem? What is the difficulty?
Anybody can start, and I know David's facing that problem, and I get calls, by the
Yeah,
way,
it's,
from many angry biotech CEOs with that same question, but please.
I would just say part of it is the large-scale manufacturing that's not scaled appropriately, and part of it is the safety certification that takes a number of months. The actual manufacturing of the cells, once you have the virus, is a matter of usually a few days, except if you have to expand the cells in culture. So it's really bringing to real scale virus production, which is a biological process. It's not a manufacturing process per se. So you have to have large amounts of cells, large amounts of media, large amounts of purification processes. And that's just, as everybody said, just at the Model T stage of development at this point. But the good news is, because there's now a margin, that is, it's a drug, it's now clear that manufacturing's companies will put effort and money into improving the process. So it will be improved, I'm sure.
Jeff, go ahead.
And maybe I can give one specific example. When you're testing viral vector, one of the things that you need to test for is these are basically disabled viruses, so they can't reproduce. They can only insert their genome into one cell. And to test that there's been no recombination event that would allow them to reproduce in the patient, you have to do a recombinant virus test. The state of the art for that test right now is to culture a sample of your product in a permissive cell line, passage it a minimum of 5 times, then take that supernatant and put that into an indicator cell line and culture it. That test takes 3 months to execute. Typical turnaround time is about 4 months by the time you set it up and report the samples. This is a place that is just primed for new technology to come in and with sequencing. There's no reason that we couldn't have results in a day or two instead of 4 months. But it takes a long time to build the data set to convince the regulators that you've got a really robust assay to assure safety.
Marcella, you wanted to make a comment?
I was also going to say that right now there's a lot, because it's so new and because 10 years ago this was science fiction, there's a lot of rules around what the potential safety issues could be. But I think that as the field is growing and as we're getting more and more data, for example, there's never been a replication competent vector. And so for everyone to spend time and resources and money doing that perhaps doesn't make sense anymore. But because many of the rules and regulations were developed for drugs and because there was an innate sort of fear of this new process, there were a lot of things put in place which I think now need to be addressed. And we know now that that's not one of the main things, and so we shouldn't be chasing that so much.
Very well said. I mean, we have several challenges. One of them is the quality standards and the requirements for other kinds of medicines are being applied to these new medicines and different steps that are creating an inordinate amount of testing. Number 2, as we heard from the panelists, the tech transfer process from an academic lab to an engineering lab is not robust. A biologist doing something in a small beaker is not really thinking about large-scale production. And I think that's normal. But that's where the state of the industry is. I know, Shashi, you wanted to make a comment on this one, and we'll take another question after that.
Yeah, I guess just to bolster your point, there's a need to bring in automation into early stages of discovery, and that doesn't exist at this point. And so that's something we need to do so that the transition and scale up from the early stages can be more directly facilitated to get higher yields and higher efficiencies.
But I'm deeply convinced that the right people are talking. You see several of them on the stage. The regulators are talking to us very closely to figure out how we get more and more of these therapies to patients as fast as possible. Let's take another question. Please.
Right, we have a question here, actually.
Okay. Actually, she had her hand up first. Sorry.
Hi. You may have addressed this already, but it's very hard for me to hear you with the echoes. So I'm curious about the bottlenecks and the relative importance of them. You've mentioned several. I'm particularly interested in to what extent are you seeing the kind of cross-discipline, cross-institutional collaboration, information sharing, resource sharing that you would like to see, and to what extent does competition get in the way and slow things down?
Excellent question.
Yeah. First, I'll give you an additional reason to be optimistic, because we've talked a lot about challenges, but the fact is that Novartis and Kite, and there are now 40 companies in the space. Novartis has run 2 global pivotal clinical trials at 25 sites, 10 countries, 4 continents. Collected cells from patients in Australia and Japan, sent them to New Jersey to be manufactured, and then the patient in Australia, Japan, Europe, they all got their cells. So I think the logistical hurdles, the manufacturing hurdles, they can be overcome. Now, you asked about what industries, what sectors come together. I think that was the sense of your question that I heard through the echoes. What I'm seeing is that companies In the automation space, companies in the technology space, I think we're going to see influence of microfluidics and robotics entering biologics so that we have technologies that can isolate, expand, grow, deliver genes to these cells. That is happening where we have cross-fertilization amongst different technology sectors. So we have so much leverage that we can take from these other technologies to improve the generation of these therapies and also the generation of the viral vectors.
I think one example is what you see on the stage. We have academic medical centers represented, 2 of the leading practitioners, actually 3 of the leading practitioners. We have an industry represented, we have an engineer represented, we have suppliers represented, and maybe we should have large pharma and regulators and payers represented as well, but it's a good start. People are working together, people are working across institutions, and we know what we are up against. And I think that makes me quite optimistic. Anybody else want to add from personal experience?
I'll just say from a supply side, for our company, it's really been enlightening for me because we have a media component of our company, and we have the tools that were made for monoclonal antibodies, but how do we apply them into this new area? And it's when we internally and with our clients start having these discussions that people start to get excited. And to be honest, this was a space that was a little too risky for a lot of big companies for a long time, but it's not anymore. So that's the sort of thing that occurs where there's investment and there's effort, and we have new people enter into the space that really add a lot of capabilities and color into the space.
I was just going to add two components, which is the academic company collaboration. It's been fantastic from our standpoint with really free exchange of ideas and processes that allowed us to go much faster than we would have otherwise in our trials. And there's another part of collaboration, which is interinstitutional academic collaborations. You know, institutions have been known to compete with each other. But I'm a pediatrician. We pediatricians always get along. And we had, in our last trial that was just published last week in New England Journal, we had children from 10 countries involved in a trial of 17 patients. And actually, you know, just 5 o'clock this morning, I had a Skype call with colleagues in Melbourne and Sydney, setting up the next collaborative effort in another rare disease. So that's another part of collaboration that this field actually has been really good at.
Thank you, everybody. This has been so inspiring. And for those of us who aren't directly in this field and are technologists and so forth, I'm wondering, when we meet again next year for Future Forum at HubWeek, what technology do you wish the room to be working on to help you in each of your labs that is an adjacent technology, or it's something like the equivalent of battery storage, make solar better? You know, like, what can we be working on to help you in your work?
Start this time, Marcella, with you.
Open to listen to the others. Well, for one thing, things that could help with chain of custody. So, for example, barcoding cells so that we know that the right patient is getting the right cell product. Right now, there's patient identifiers on there, which is reasonable given the risks of giving the wrong patient the wrong cells. But it would be useful, I think, to have a system like that. Systems that could process blood at a closed system. So right now, part of the purification of cells and getting vectors and cells ready, there's open stages to it, which means that you need to have a room with environmental monitoring, and it has to have has to have particle counts and so on that would be FDA compliant. But if you can do things in a closed system, then it makes things a little bit faster and a little bit better, and you don't necessarily have to have that level of environment control if you can keep everything in the bag without opening the bag. So those are two things I can think of off the bat.
Else want to add?
Yeah, I'd add to that automation, things either at the unit op level or the integrated level, but bringing automation and ease of data portability and analytics I think this is a field that's ripe for big data. Every patient that comes in is a different starting material with a different history, with a different genome. No two patients are going to be alike. And when we treat the patients, the cells grow in the patients. So unlike a traditional drug, you can't characterize what's in the vial and think that that's the end. It's going to change when it's administered. Understanding that and how that impacts the success of treatment is going to be critically important. And this is such a complex equation that people are going to have a very hard time figuring it out, with pencil and paper. We're going to need to apply some really new data tools to really understand this space.
I would add to that, if any concepts related to applying machine learning and image recognition to cell culture would be enormous.
I think you've heard the very pragmatic to the idealistic, and I think there are pragmatic answers that we need to have so that there are more viral vectors being produced, so that we don't have a year waiting time or you don't get a slot for 2019. I think that's a very pragmatic problem that we have today. And there are more detailed technological challenges or technological solutions of creating a closed system, a bedside system. And I think we have people working on all of those problems at this point in time. This has been a fascinating panel, and I want to close by firstly thanking all the panelists. They've come from many different locations. Thank you very, very much. It's really an honor to be on stage with you guys. It's a rarity that we get all of you together. You would agree with me that there is a lot of promise in cell therapy. We should not get too idealistic and assume that it's going to be available for primary care tomorrow. Yet, advances are being made, and really smart and dedicated people are working on it. I do expect that in a year's time, we will have made some significant advances with the number of people who are working on it. Yet, there are challenges. We talked about a few of them today, but I'm super optimistic that in the next few years, we will see this therapy really changing how medicine is practiced. I want to thank you again for your attention, and thank you for joining us.